Hi, my name is Yaoheng Yang. I am from Professor Hong Chen's lab. It is my great pleasure to share my recent work in the FAST Symposium. The title of today's presentation is TRPV1-Mediated Sonogenetics for Non-Invasive and Cell-Type Selective Control of Neurons, Neural Circuits, and Mouse Behavior. Sonogenetics is the technology that combines the ultrasound and the genetic tool for non-invasive and cell-type selective neuromodulation. First, the wild-type neuron is genetically transfected by the viral vector to express ultrasound-sensitive ion channels to the specific neuron type. And these ultrasound-sensitive ion channels can be opened by ultrasound to activate the neurons. And in this way, the transfected neurons are more sensitive to ultrasound than the wild-type neurons. So when ultrasound is applied, only the transfected neurons can be activated, while the surrounding wild-type neurons will not be affected. The existing sonogenetics are all based on the mechanosensitive ion channels or proteins by harnessing the mechanical effect of ultrasound. Sonogenetics was first proposed in 2015. The mechanosensitive ion channel TRP4 was demonstrated to be activated by ultrasound in C. elegans with the help of microbubbles. Recently, 3 milestone papers reported successful sonogenetic neuromodulation in mice based on Prestin, Mastral, and the TRPA1 ion channel. And divergent from the current mechanical paradigm, we proposed to use thermosensitive ion channel TRPV1 to develop sonogenetics. We hypothesized that TRPV1 is an ultrasound-sensitive ion channel. And as illustrated here, we expected that neurons genetically transfected to express TRPV1 can be activated by ultrasound-induced mild heating. And the objective of this study was to develop TRPV1-mediated sonogenetics. And this objective was achieved by 3 studies to demonstrate the capability of sonogenetics to control specific neuron activity, neural circuit activity, and the behavior change. First, I would like to introduce to evaluate the feasibility of using sonogenetics to selectively control neuronal activity. We first designed and developed a unique system that combined in vivo 2-photon microscope with the ring-shaped fast transducer to directly image single-cell neuronal activity in mouse brain during fast sonication. And as shown in this illustration, the ring transducer was coupled with the objective of 2-photon microscope to align the fast beam and the optical beam confocally. And by creating a transparent optical window on the mouse skull, 2-photon microscope can direct image neuronal activity in a living mouse. And this is a representative 2-photon image of the mouse brain in vivo. The ultrasound focus covers the whole entire field of view. And the red color represents TRPV1 that was transfected by the virus injection. And the green color represents GCAM, which is a calcium indicator that can indicate the activation of neurons. And we can identify 2 types of neurons here. So like neuron 1 and 2, they both express GCAM and also express the TRPV1. And the other type of neurons, such as neuron 3 and 4, they only express GCAM, but not the TRPV1. And by analyzing the calcium curve over time, we can clearly see that only when the neurons that overexpressing TRPV1, it can be activated by the ultrasound stimulation as indicated by this yellow bar. While the surrounding wild-type neurons without the overexpression of TRPV1, there was no response to the ultrasound stimulation. And these results indicate that the sonogenetics can achieve cell-type selective neuromodulations. And to my knowledge, that all previous sonogenetic studies provide indirect readouts such as EMG, EEG, or behavior change. And we, for the first time, showing direct evidence that sonogenetics can achieve cell-type selective neuromodulation in vivo. And to further confirm the result, we performed the experiment in multiple mice and receiving multiple sonication. And we confirmed that only when the neurons overexpressing TRPV1, they can be activated by ultrasound. In contrast, the wild-type neurons without expressing TRPV1 had no response. And based on these calcium signals, we quantified the success rate of neuronal activation. And the TRPV1-mediated sonogenetics can achieve success rate as high as 75%, which is significantly higher than the control mouse. And after sonication, we performed the safety evaluation by histologically staining the neurons, astrocyte, microglia cells, and apoptotic cells, and we did not find any evidence that showing the ultrasound caused any damage. And after demonstrate the feasibility of TRPV1-mediated sonogenetics to control neuronal activity, we then investigate whether sonogenetics can activate the whole neural circuits. And we use the functional PET to image the whole brain activity during sonication. Here shows the representative image from two groups. The left one is sonogenetics group, which is the TRPV1 positive mouse. And the right one is the FAST alone group in wild-type mouse without TRPV1. And we can see that FAST can successfully activate the brain region at the target location in TRPV1 positive mouse compared to the contralateral location. In contrast, this activity is not observed in the wild-type TRPV1 negative mouse. And we also performed additional control groups that without the fast sonication. And in these two control groups, the brain is symmetric between left and right. And there was no signal enhancement at the ultrasound target location. And in addition to the activation at the target location, we also observed the change of the global network induced by the sonogenetic stimulation. And this result demonstrated that sonogenetics is able to activate targeted brain region and as well as the associated regions. And combining sonogenetics with the functional PET could also be a powerful research tool to investigate the functional connectivity of the whole brain. And the last study evaluated the TRPV1-mediated sonogenetics control of the locomotor behavior in freely moving mice. And we designed a wearable fast transducer to the mouse head to sonicate the striatum in freely moving mice, which is the brain region that controls the rotational behavior. And here shows the behavior response of two mouse. The left one with TRPV1 overexpression, and the right one was injected by the control virus without TRPV1 expression. And the FAST can evoke the rotational behavior only in the TRPV1 positive mice, while there was almost no effect in the control mice. To further verify the result, we performed the experiment in multiple mice, and each mice receiving 3 or 5 repeated sonication. And in TRPV1 positive mice, 33 out of 38 sonication in 1.3 megapascal group showed successful stimulation and evoked rotational behavior specifically in the contralateral direction. And by reducing the acoustic pressure, both the success rate and the rotational speed was also decreased. And in contrast, there was almost no response observed in the control mice without TRPV1 expression. And this result demonstrates that sonogenetics can evoke circuit-specific locomotor behavior in freely moving mouse. To conclude, we developed the TRPV1-mediated sonogenetics, and we provide direct evidence demonstrating that sonogenetics is able to selectively activate neurons in vivo. And by using functional PET image, we proved that sonogenetics is able to activate neural circuits. And last, we show that sonogenetics stimulation at striatum can evoke circuit-specific locomotor behavior in freely moving mice. And I would like to thank our whole lab, my great mentor, Dr. Hong Chen, our collaborators, and also the funding agencies. And thank you, everyone.